Good afternoon, everyone. Thank you for coming. Today we have with us Ayelet Landau from the psychology department here at the Hebrew University. Ayelet received her PhD from Berkeley University, studying cognition, brain, and behavior. After her PhD, Ayelet headed the Ernst Strungman Institute for Neuroscience in the Max Planck in Frankfurt. Ayelet and her group study cognitive functions related to attention and timing, and today it will tell us about the research for internal pacemaker in the brain and how mind measures interval and how it can study cognitive processes that underline temporal decision. Ayelet, thank you for coming.
So thanks, Jonathan, for the introduction. It's great to be here. I presented here a few years back, and it's fun to come to this forum and to talk about actually a completely new talk. So this is the first time I'm giving this talk. It's mostly work in the last 2 years, where my team over in Mount Focus and I have been focusing on interval timing. So what is interval timing? It's really a fancy technical term to say time perception, but if I say time perception, sometimes people start thinking about where they were in first grade or what happened, in the 3 waves ago of the COVID pandemic. So interval timing is really time perception in the seconds intervals. And you might wonder, why is this relevant to your everyday life? And for this, actually, in a discussion with Landau and ELSC, we came up with a really good metaphor that really helps understand why this is important. So obviously, timing is crucial in many, many things we do with our senses and with our motor systems. But of course, one of the feats of cognition are the ability to prepare proper popcorn, which is really reliant on your ability to kind of tune in to some statistics that are happening with the pops of the popcorn. And then at some point, you reach some kind of threshold where you say, okay, I must stop. Otherwise, this will go very, very badly. So this is the metaphor I would like you, or the analogy I would like you to kind of hang on to as we go through the talk. But of course, it's only one metaphor, and there are many ways in which timing is very central to our functions. In my talk today, I will answer 2 main questions. I will ask, is there a role for sensory processing in interval timing? And the second question will address whether we can identify neural signatures for interval timing. And the context of these 2 questions are basically a model that I would like to propose for timing, which is hierarchical. So I'll just pay quick tribute to what has been written about models for timing in the brain. John Schlurf and Rich Ivory in 2008 put forth an opinion piece where they tried to classify the type of timing models into 2 variants. One variant is an intrinsic model, and that's kind of a fancy word to say anything your brain is doing has a price tag in timing. So your subjective timing is made of basically any expended energy that your brain is busy with. And on the other hand, there's another camp talking about dedicated models. Those would be models which typically, it doesn't have to be that way from a purely analytic standpoint, but typically they postulate that in the brain we have some clocks manifest in some structures. I highlight here the cerebellum, the hippocampus, the basal ganglia, as well as frontal cortex. All of these loci have been proposed to do this timing, perhaps accumulate some form of abstract tics. I will say that the evidence for either camp are not compelling, and I'm not going to go into the issues I have with each and every possible type of evidence provided for each model, but I will try to propose that, as is the case often when we have 2 kind of camps in a debate, it's possible that we have evidence for both type of timing processes in the brain. And so I would like to propose that timing is achieved by a hierarchical structure, if you will, where we have a system-specific contribution to the process of timing, but we then also have domain general processes that help us do this cognitive feat. And in my research, and this is kind of a summary, if you will, of various research that is going on in the lab, we try to characterize both the sensory-specific engagement in timing as well as the domain general processes. And ultimately, we also hope to be able to achieve access to measuring both at the same time, potentially trying to understand how sensory signatures of timing might then be integrated to domain general modules for time. That is the theoretical framework, or that is the kind of roadmap of the general research, but I'm going to narrow down now back to the two questions I plan to ask. And we'll start with looking for those sensory evidence for timing using primarily behavioral methods. So it turns out timing is not like a fixed clock, okay? Both our group and other groups have shown that if we do present two objective intervals, quite often there are non-temporal manipulations we can do to the stimuli that is being timed that will alter the way we perceive time. So you watch a kettle boil, it never boils. That feels like a much longer period of time compared to, for example, if you divert attention to something else while that same interval has to expire. You look at the stronger stimulus, it seems to last longer than a dim stimulus, and finally, the stimulus that is moving also seems to subtend more time than a stimulus that is static. As a first step in understanding whether this is a general rule in timing, that stronger stimuli creates a longer perceived interval, we turn to another sense and asked, can we also see this kind of finding when we look at the tactile modality? So in the first study, I will show a very simple study examining whether tactile intensity alters our sense of timing. It will be kind of a way also to present the basic methodologies that will then also repeat as I carry on in the other parts of the talk. So a task that we often use in the lab is called the temporal dissection task. And it's a task in which we teach our individuals, the participants, to reference intervals. In this case, half a second and a little over 2 seconds. They learn what's long, they learn what's short over very few trials. And then we go and head over to the main experiment where we will have several different intervals. And our individuals will still have to do this classification. Of course, the classification becomes a little bit more difficult. If we look at their performance, obviously when we present the reference stimuli and we plot it as percent long on the y-axis here, they perform perfectly. If it's short, they always say it's short. If it's long, they always say it's long. And we're interested in the dynamic range in between. How do they perform on those in-between intervals? To this type of data, this is just a single subject data. We can fit a function. Several functions are out there that would do this job. And when we have a function now, we could go a little bit beyond the observable data and say something about that very special point, which is the point where people are 50% saying long. So if you will, the point of confusion. They're as likely to say long as short for the same objective interval. And we call that point the bisection point. So when we plot the data like this, this was actually, as I said, a single subject data, and it was actually in response to vibrotactile stimuli that were of a weak intensity. I now will add the data for the strong intensity, and the general thing we look at when we look at these type of data, for those who are less familiar with these data, is a shift to the right or to the left. So if I see a shift to the right, that means that I needed to present the stimulus longer for it to be perceived at the same duration. So there is an underestimation of time. While if the function is shifted to the left, that means there's an overestimation of time. I can reach the same subjective sensation with less time because time is expanded. These are the data for this particular subject. So this particular subject shows a very clear effect showing that when you present a strong stimulus, time seems to be estimated as longer than when you present a weak stimulus. I'm going to show off with some individual subject data with a scatter plot, which plots strong vibrotactile stimulation bisection point over weak vibrotactile stimulation bisection point. And basically, if there were no effect at all, we'd expect dots to be on the unity line. If stronger judgment is longer for a given participant, we expect it to appear up here. And this is my entire sample. So while some subjects have a huge change in their decision, while others have a smaller, they all basically show this effect. So on the face of it, stronger stimuli in the touch modality seems to last longer. It's a potential piece of evidence for the role of sensory systems. So maybe we can say that interval timing is made out of our perceptions or our sensations. Of course, a stronger stimulus feels very different from a weak stimulus. So I will just withhold that conclusion and try to show you some extra evidence that might be even more compelling case for sensory engagement in interval time. So in the next study, led by Dr. Shani Grossman and Chen Guetta, Dr. Shani Grossman was then a PhD at the Weizmann Institute, and Chen Guetta at the time was a master's student in Lyon de Wellsbach, now a PhD student there. We asked about what happens with time estimation when we lose sensory input. And we were interested in the case of blinks, in this activity that we're all engaged in every few seconds. All of you are basically experiencing a complete blackout of your sensory inputs due to the covering of your eyelids of the eye. We know from other studies that if we lock brain activity and measure, for example, bowl response and other responses, we see a big decrease in visual cortex. However, and this video, courtesy of Tal Golem, shows obviously the obvious blackout that you can all evidence here, but of course the person blinking there is not aware of the degree of blackout that this movie is undergoing. Okay, so that's just a demonstration of how vast the loss of sensory input is. And we like to think of this as an ecological illusion, right? Because basically you're not doing this in any voluntary sense. You're not even aware of it, but your visual cortex has just experienced a huge drop in processing. So is timing affected by this involuntary, unaware loss of visual input? Was what we wanted to know. So we headed and measured interval timing with and without blinks. The task is very similar to the task that I just presented to you. People are making a visual temporal bisection task. They're judging whether this disk is short or long. These were the intervals, 0.6 to 2.8. These were the reference intervals. And of course, we tasked them with the judgment over all different intervals and get their performance. As before, we will plot it as probability long responses as a function of the interval duration. And this is just the group data showing the expected pattern. But what we then did is we took all the trials and we classified them to those in which a subject has spontaneously blinked during the interval and to those in which the subject did not blink. And yes, this is the bisection point. This is the measure we typically compare. As before, a shift in one condition to the right of the other would mean that the one on the right is underestimated compared to the one on the left. So these are the data. And as you can see, a blink-free interval is just as longer than a blink interval. Okay, so when we blink, we lose time. How much time do we lose? We looked at the data at first and saw 120 milliseconds. We looked at then the blink duration and thought to ourselves, that is very similar. The blink duration, the mean blink duration was also around 120 milliseconds. So we thought this is quite compelling. The time that is lost is actually the time that we measure our subjects are in darkness. But of course, we could also do a little bit better because maybe I blink really slowly, a very slow, juicy blink, and Eli can't miss a moment, so he really blinks very, very fast. So maybe I lose so much more time than Eli because, he's Eli. And so we basically quantified the individual subject bisection point and the individual subject's blink duration, and we find a moderate relationship between the two, which is significant. So this is kind of compelling evidence, which I think help us adopt the interpretation that indeed sensory processing has a role in interval timing. But of course, I can't fully say that before I convince you that this is vision specific, right? Because maybe a blink is like this crazy brain hiccup and the entire brain basically shuts down for a moment. And then again, I'm back to some either domain general or global mechanisms. And so in order to rule that possibility out, we ran the same experiment in the auditory domain. People were judging intervals using audition, and we classified trials to blink and blink-free trials, and we do not see any modulation in time perception under these conditions. So, is timing affected by involuntary, unaware loss of visual input? Seems to be the case, yes. When we have our eyes open, we are subjectively experiencing more time than when we have this spontaneous blink. A small note. People study blinks. It's not a huge community. The community that studies spontaneous blinks is even smaller. So that was a little bit of an extra feat, which took some trickery. Ask me later how we did it. But basically, these blinks are all involuntary, and that's really
Can you ask a technical question?
Absolutely, please.
The differences that you have in the distribution of the effect could be related to the duration of the blink? Because there are many trials where the subject did not estimate at the time as if he didn't have any blink. In some of the trials, he had blink, but the duration is just like without blink. The data is distributed, right? You see the correlation of 0.47.
Right.
Not 1.
That's true. So there is noise
So
in
if
our
you
measurement.
measure the duration of the blinks, are they also variable? And
I'm
could
sure.
you correlate these durations to the change in the distribution?
So you're saying go within an individual and look at the variability
The
and.
individual blink.
So this is the scatter plot looks at the different subjects' blink duration and correlates it with the shift in the bisection point. It's important to kind of note the sources of noise when you do these type of quantifications, because basically this is a camera-based measure, and there's a certain criteria of when do we call something a blink. So the measurement is not the most clean measurement. So at least in our field, 0.44 or 47 correlation, which happens in this sample to be significant, is a decent demonstration.
I'm not arguing against the validity of.
So you're thinking there's another source of variability that could be inquired about.
It explains the process mechanism.
Could you repeat?
How important is the length of time that you miss this information that comes from the eye?
But I think the idea that Elon is saying.
Different mechanisms than, general attention or technical.
Omission.
We can talk about
Yes, it sounds interesting. I'm always in for sources of variance that I can explain, so you will have to explain to me.
So it's a little bit related, but your task here is not really measuring time, it's integrating stimulus over time. So if you shut down the stimulus, it doesn't mean there's some internal sense of time that changes. You just don't integrate it over the stimulus.
So you're not interpolating, at the very least, right? So this is true. I mean, I think this is a relevant phrasing of the process, and I think that the rest of the talk is going to get closer to this type of description. So we can maybe carry on and then supplement with exercises in the rest of it. So at this point, I'm happy to provide this as further support that interval timing is at least partially contributed by sensory processing. A little bit along the line of what Jonathan just kind of raised, I'm not sure if that's what you meant, is there are models out there for interval timing. And one particular model that we are turning to at this point in the talk, and that will also potentially be useful for the rest of the talk, are drift diffusion models. So when we're timing, and this model provided by Simon and Bachi is a model that targets specifically the bisection task, the temporal bisection task. Their model is a little bit more complicated, but for our purposes, a simplified version of their model works just as well. And the main idea in these models is that evidence is being accumulated. Time is being accumulated. And in the task, there's also a threshold. So the time is being accumulated to a boundary or to a threshold. And in a given trial, we can symbolize the process of accumulation with this wiggly line. And if the interval ended before the wiggly line, accumulated evidence made it to the threshold, we will classify it as a short interval. If, on the other hand, the evidence is accumulated and it passes the threshold, we will classify this trial as a long interval. And of course, over the entire experiment, we will have several repetitions of trials, and we can deduct some parameters from this model, such as what is the rate of accumulation and what is the noise that we have, which is also scaled with time in such a process. this model does a decent job in quantifying the probability to reach the threshold, which plotted in an accumulated fashion looks very much like the functions that I've shown you before. So technically this could be a nice cognitive model that could account for our psychophysical data. And now, if we go back to the studies I've just shown you, we can try and think, okay, so what does this mean about the process when a stronger stimulus, for example, is experienced as longer? So if this were my weak tactile stimulation, it is possible to think about the process changing in the following manner. For example, the rate increasing with a stronger stimulus, leading to a faster crossing of the boundary, which we interpret as an extended experience of time. So that would be kind of one way to look at the process underlying what I've just shown you. And in the case of the blinks, once again, we have the process unfolding to a boundary, and when we blink, at the very least, there seems to be no accumulation of this quantity of time as we conceptualize it. So it seems to be a useful model to think about these type of data, and it will become quite instrumental when we turn to trying to find neural signatures of the process of timing. And specifically, I should say, these models have been fitted for, or constructed to fit behavioral data, both accuracy and reaction time data. This specific model does a good job for the accuracy, as I've shown it to you before. But when we turn to neural data, we can actually try to gain access not only to these type of, this type of representation, but perhaps we might be able to even say something about the fashion in which accumulation occurs in interval time. And in order to address that, now or later, maybe later, okay. In order to address that, I would like to turn to the work of Neil O'Phil, who is an ELSC student in my lab. Neil actually approached me after my previous ELSC talk, and it's worked out pretty nicely. And so the next series of studies are a portion of what he has been up to ever since. We ask, can we find a neural signature for interval timing? And we turn, of course, to the temporal bisection task. So we turn back to the temporal bisection task, and we do it in the visual modality. We use gratings. We really like gratings. If you've seen my previous ELSC task, you might know more. I'm happy to elaborate on this. If you ever took a class with me, you'll hear all about why we love gratings, but for this talk, it's actually less relevant. We have the same type of manipulation where we have a short or a long interval as a reference. We train our subjects on those short and long references, and then we throw at them a bunch of different intervals in order to get their classification performance on this task. Neil was measuring EEG activity, so this is measurement, a non-invasive measurement measured from the scalp of basically a very far away measurement of LFPs from the brain. And what I'm showing you here are data from the offset of the interval. So if we consider these type of stimuli, the visual stimulus, for many of the intervals, it is going to be the offset of the interval, which is actually a really important moment, right? That's the moment where it's time to start or plan or decide about this interval, whether it was long or short. And also from a sensory perspective, I do come from sensory systems. It's a nice moment because you typically have a very nice lineup of the physiological response. So 0 here is time from stimulus offset. This is a diagram, a schematic diagram of the scalp topography measured at the group level. Y-axis in these measurements are the microvolts that are measured from the sensors. And specifically what's highlighted in this topography is the fact that we're averaging over 9 channels. So you can see that there is some action going on right after the offset, but then there is a very large potential. What I'm plotting here is the 0.5 second interval, and I will now gradually add the longer interval. So adding 0.7 seconds, then 0.9, then 0.05, and when you just look at these intervals, what seems to be happening is that we have a response that seems to track duration, right? So the shorter the interval is, the larger this response is, and it then becomes diminished as the interval becomes longer. Except we add all the intervals, and you can see that at a certain moment in this progression, this response saturates or diminishes, depends how you want to look at it. And now we're thinking it's tracking duration, but perhaps it's also tracking our bounded decision. So this, of course, requires a little bit of a closer look, and in order to reduce some dimensionality, I'm going to be representing these data by taking this fan window, which is 200 milliseconds, and I'm plotting it now with our good old durations axis as the x-axis, and the magnitude of the response is now, the microvolts is averaged from these 200 milliseconds. So you can, again, appreciate this kind of linear decrease with duration, except that then this response saturates.
Can I ask a question?
You can.
If they're not in a task, and you show them these stimuli, what do you get?
I did not measure that, but I think it's a very interesting question. And I think these are the type of questions that we, so there are tons of questions that this figure raised. I will answer, I think, 2 follow-up questions on this, but I think this is a question that we could seek experimental evidence for.
Yes. Already at point 2, there is the responses that are correlated with the duration. Is this significant, or is this, or you ignore it because it's?
Yeah, I'd be happy to comment on it. It's true. There is some dependency in duration in this window as well. I'm going to show you some extra evidence, and then ask me that question again, because I think this element has a story of its own, which I'm happy to go into as well. Yeah.
And also, is there a dependency of the late response and this early response?
That's a good point. I think, again, I will have a partial answer to that after a couple of extra experiments. Okay, so these are the physiological.
When is the reaction time? When is the chocolate?
You want to see the behavioral data. So there they are. The reaction times have a dynamic of their own. I can comment more about it, but it's not the focus of what I'm showing here. Generally speaking, I plot the data as I plotted it before, percent long response as a function of stimulus duration. Reaction time dynamic in these tests have, just briefly I will say, you have some kind of baseline reaction time for the easy ends of this plot, right? So this is an easy trial, and this is an easy trial. And over here it gets a little bit hairy, gets a little bit difficult. Is this long, is this short? And you typically will see evidence for that in the reaction times. So you'll see an increase in reaction time. We don't always see it, but if there's any trend in the reaction time, you typically will see that there's a reasonable baseline reaction time, then a slight increase in reaction time, then back to baseline for those easy trials.
I'm just looking at the left graph.
Yes.
You're putting a spotlight on 300 milliseconds to 500. My question is whether the subjects already made the decision by that.
It's a very good question. I have more extra slides to speak to that. But I would like to first describe the basic phenomenon, show you the approach that we took, and then please ask these questions at the end, because I do have things to attest to that.
But it's 300 milliseconds, so we assume that if not all, most of his decisions were made.
So this is, I think, what we were investigating. So this talk could have had the title, I thought I'm studying perception, I ended up studying decision making. For sure. So that's definitely what's going on here. And we're trying to describe this decision process. And I think that what Neil in his PhD has discovered is a really good handle in a physiological response. And in a second, I'll also show that we have a nice formal description, which is simple and useful. I'd say elegant. I hope you agree with me. So these are the data. And remember that group diffusion model that we discussed before. And remember that I kind of put a pistol in the first act saying, if I'm doing physiology, maybe I could take something from this rather than just kind of summarize probabilities of crossing some boundary. So you could basically take the same framework, but instead of quantifying, indeed, the probability to cross a boundary, we chose to focus on the distance to the boundary, because now we have a continuous measure. I mean, maybe it's not an online continuous measure, but it is a physiological measure of a continuous process, which we still assume happens in the brain, okay? So I'm highlighting now the red lines because this is the quantity that we focus on, and this type of quantity produces a function that looks more or less like that. What did Nir do next? Well, the next step that he did was use this model to fit the psychophysical data, right? So I previously showed you that this is a decent approximation of behavioral data, and indeed this type of model was written, it was formulated for these type of data. But what he did next was he took the parameters from the behavioral modeling of the data, and by simulation fit these parameters to the physiological response. Okay, so if indeed we are tapping into the same process, we hoped this would provide a decent fit. And indeed, the fit was very strong. The fit was strong, and I would say if we explore parameter space of these models, the parameters from the behavior actually reach the optimal fit that this model has for the EEG data. We could do another thing. We could also ask, what happens when I have a given stimulus duration? Can this neural response predict what my subject will say, long or short? And so for that, we focus on each individual subject's dissection point, and we extract the event-related potentials for the 50% of trials in which they said short, because that's the definition of dissection point, right? 50-50 moment. And the 50% trials in which they said long, and as you can see, there's a very marked difference between those. So any concerns anyone might have about the physical durations changing as a function of me trying to describe these data, this is a moment, or these are data to physically identical stimuli within subjects. And this is the nice marked difference we see. We next turn to ask whether this is a domain general type of signature, or perhaps it is visually specific to vision. And so you might guess by now that what I basically did, we basically acquired the same exact experimental design, except instead of the visual bisection, we did a tactile bisection, same timing parameters, but this time using vibrant tactile stimulation to the fingertip. And these are the data. So they're noisier. There are several reasons for that, but the phenomenon is basically replicated. Now durations are represented here with a color bar, and as you can see, shorter durations have this stronger response, and then the response somehow saturates. This shows it a little bit better, seeing that the data is a little noisier. And again, the fits are above 0.9. When we take the behavioral parameters, or the parameters taken from the behavioral fits onto the physiological data. I will say in a word, back to the question that was raised about the earlier components, the earlier modulations with duration seem to not share scalp topography. So what I'm focusing on today, these 200 milliseconds, the topographies are very, very similar, suggesting that we're looking at a very similar mechanism. But if you do touch or if you do vision, these earlier responses seem to have different distributions. So they might point to the sensory evidence accumulation, which is then perhaps by 300 milliseconds turned into some more domain general mechanism. We could do the same trick, look at the point of the dissection point, and look at the responses. And here too, the neural response can predict for a physically identical stimulus the behavioral decision. Finally, we also wanted to see whether there was something special about the intervals that we used. Maybe offset responses after a stimulus was up for 0.5 to 2.1 seconds are kind of some lucky streak of adaptation processes that happen to saturate, right around where our dissection point was. Right? This is not what I was proposing in interpreting the data so far, but I think it's an important thing to say. If we're looking at a bounded, if we're looking at a process that is quantifying the distance to a threshold, what happens if we change the threshold by changing the statistics of the task? And so for that purpose, we ran within subject two separate blocks, one in which we had a sub-second time range, 0.2 to 0.8, and another with 1 to 2 seconds. And the block order was counterbalanced, and we added some extra other controls that if someone asks about, I'll point back to this experiment. But this is the main manipulation in this experiment. And I'll start by showing the data for the 0.2 to 0.8 time range. First of all, this is another interpretation of the finding. We have less intervals, but once again we see that the parameters that we take from behavior fit the data very well. I didn't put all the R-square values here, my bad, but they're all about 0.9. And now let's turn to the supersecond intervals, 1 second to 2 seconds. Oh, we do the same trick. We look at the point, dissection point, and we see the same pattern. And now turning to the 1 to 2 second interval, we basically see the same process. So this is within subject, and I think this helps us say with above, it suggests that the model that we're thinking about, where this response basically expresses the distance to the boundary, to the threshold, seems to be reasonable. We move the threshold, and the dynamic is replicated towards this new threshold. I think this figure needs no further explanation. So this would be a good moment to ask questions about these findings.
So what is the response that the subject needs to do? What type of, is it verbal? Is it like pressing a key?
Right. I should have known, that's exactly why I said someone will ask about that control, and I should have actually said you would ask about that control. So people are pressing a button. In the first visual experiment, they were pressing a button with one hand. In the tactile experiment, they were pressing a button with one hand because we were using their other fingers for stimulation. And in the last study, Neil did a very smart thing. He basically had half the subject say long and short, and then had the other half of the subject say long and short. So counterbalance the effectors to make sure that this is not sensitive to the either hand of use or that it goes beyond the preparation of a specific motor plan.
But can't you tell the same subject as well?
Sorry?
The same subject as left and right.
You would want us to further switch within the subject. We chose to do it differently, but that would be also reasonable. I mean, I personally, this is like experimental intuition, I would be concerned about carryover effects and noisy data, but I think it could.
But generally, you think there's no relation to motor response?
So I think that the evidence that we see, and I reduced the data obviously, but we look at distributions, and we, and of course everything is backed up with heavy GLM analyses and significance, but what we see is that it doesn't matter if you're planning, if your long response is the right hand or the left hand, and it doesn't matter, I mean, basically the distribution is...
Some places in the cortex would care about this, right?
Sorry?
Somewhere in the cortex would care about moving the right hand.
Sure, yeah, of course. We have effector-specific responsivity, but when we do a GLM, we model actually duration by decision interactions. And then we're basically modeling what I try to give a more direct intuition by looking at the data and by summarizing it.
But in terms of time, from 0, when is the response taken?
The responses are typically after half a second.
After half a second. So that's a very long reaction time. I mean, so we usually have, I don't know, between 3 to 4, and even half a second.
This is not a simple reaction time task. It's a cognitive task.
What I'm concerned about is that a large part of the response that you see are actually related to the motor plan. Not
for you, I have an extra slide. Should I put it up now?
No,
why not? So I think, this type of, I think it's kind of a flavor, right? I am a perceptual researcher, so for me an offset is super exciting. I feel like I've locked to some ground truth, and it makes me very, very happy. But to be honest, I'm not attached to this perspective. And I think that this particular component and dynamic can readily be described if we lock to the response. And so that's what we did, because we thought someone might want to know whether or how it looks when we lock to the response. So this is some excruciatingly painful supplementary figure, but you could just, and I'm not sure we have all the information for you to decipher it, but if you want, you can focus on this panel which would be the first experiment, visual with durations from 0.5 to 2.1. And you can see what this dynamic looks like locked to the response. And here you have the two sibling blocks with the short subsecond and the long suprasecond responses. This dynamic is very, very similar. And the model will work with these as well. So, and I think it's important. And when I write that paragraph in the discussion section, I propose to the literature that these are two different angles to look at the same process. And it's an interesting, I'm not sure how consequential, but it's an interesting question to think about whether there is a right way to look at this phenomenon.
But maybe you said it, but did you talk about which electrodes were included in each analysis?
Yes. So I highlighted at the first data figure the 9 electrodes we used. And we have really cool topographies. Do I have the cool topographies? So I'll show you a kind of simple topography, and I will verbally walk you through an imaginary topography that I don't have to show you. So this is the very simple topography highlighting which electrodes we're looking at, and as you can see, this is quite a distributed dynamic. What we are providing our kind of reviewers these days is a topography of the fits. And the topography of the fits are quite beautifully centered here. Why these 9 electrodes? Honestly, we do very sophisticated cluster analysis. We could have used many more.
No, but there are many specific questions to ask, depending on which electrode you use.
So,
you cannot just
right, no, I showed you that these are the 9 electrodes we're looking at, and they are, like, no matter which statistics we use, those 9 electrodes seem to be part of a relevant cluster.
In many senses, they are, for you as a perceptual person, they would take a little bit back, and for me, a bit.
I guess I'm going to have to.
It's kind of
I'm going to have to
completely motor.
give up some of
This is
my
really the
perception.
heart of the motor area of this person. Yeah.
So this is group. This is the group topography, and I think there are people out there that are willing to sign off a theory saying that timing is all manifested motor. Like that opinion is out there. I hesitate to use EEG to kind of sign off a particular substrate, but I think we've done a decent job using EEG to describe a cognitive process, which is formally described in a model.
The rest of the comments are for discussion. But
Okay,
I would
so-
love to talk to you after the seminar
Okay.
about possibilities. Okay.
Sounds good. So I'm going to summarize. These are the questions I wanted to ask. I'm going to outline my conclusions for today. This is an ongoing project, but I'm going to outline the conclusions for today on the proposed hierarchy that we are attempting to examine. In the first part of the talk, I've demonstrated that intensity impacts the senses of the sense of time in vision and in touch. And I've also shown this after input loss, which is not reaching our awareness and which occurs involuntarily, which I think is a strong piece of evidence, and that's visual selective. So that's strong evidence for a role of visual signatures and sensory signatures in the process of timing. And then we've shown that we found an EEG response that reflects the accumulated evidence towards a temporal decision. It is modality independent as far as we've tested, in touch and vision. It is determined by the statistics of the task, so it's relative timing. And timing is also made of bounded evidence accumulation. So we have a computational model that we find is a very good description of the physiology that we have measured. As I said before, and I'm very curious to hear other questions, this is ongoing research, and we hope that this nice description of the signature will open the road for other investigations on the topic and maybe even sharpen our pencils in terms of what it is that temporal estimation is made of. I should also just say that coming next in the research program is to start getting at how is the information from sensory cortices then integrated in domain general computations. This is a project that is starting to, that we are finally ready to start embarking on, and will likely involve invasive measurements that enable us to measure basal ganglia responses simultaneously with EEG responses in order to try and see basal ganglia as a candidate central locus of timing. How does it synchronize? How does it integrate information from different sensory modalities? So this is, of course, in collaboration with researchers that have access and allow us access to this type of population. So I hope you'll never view your popcorn popping the same again. This weekend seems to be very suited for some home popcorn preparation. I am extremely fortunate to have a lab filled with extremely talented students, for which I am grateful. And I would like to thank you for your time, as well as the funding agencies that make this research possible. And now is the time for questions.
I want to get back to the popcorn metaphor. It's, in a sense, there are two ways in which it's different from the experiment that you were running. First of all, it was auditory. And secondly,
So you should ask, where is the auditory?
secondly, no, but more seriously. So one question is what happens in the